you, asking you about recording, please say yes. And with that way, people can view this webinar in the future. Okay, so Abe, are you sharing?
Yeah, I just
Okay.
since you brought up the webinar, so I just
Yeah,
said
yeah.
it's up here in case
Mm-hmm.
anyone's interested. Has the IOC, some things like career panel, AI,
Mm-hmm.
and various entrepreneurship, very timely and hopefully useful topics for
Mm-hmm.
both students and faculty. And then there's some of the more distinguished lectures like today's included that you can all access. So it's I think hopefully this will be of broad interest to the
Mm-hmm.
community. Um,
Absolutely.
okay, well, without further ado, I'll get really quickly started because I really want to give the time all to Shubal. So welcome everyone. This is the Bioengineering Institute of California's. It's a California Academy um, distinguished lecture, and we are very excited to have Dr. Shubal Roy to share his vision, his work, but then leading into his vision of how there's a serious problem in the kidney disease and how he's addressing it, but also I talked to him about this is a grand challenge potentially for Californians to come together, bringing different expertise together, supporting a big problem, a pervasive problem, not only California, but also worldwide. I'm sure Shiva will cover that and stating the problem, but also wanting to look for what kind of team maybe as big we can help to facilitate, come together, and maybe go after mechanisms to attack this dreadful problem. So I'll let Shiva, you introduce yourself. He's at UCSF. For many years and really focusing on this problem throughout his career. So he's the right person to really give us an understanding of the problem and some of the current state. It's all yours, Shiva.
Thank you, Abe. Thank you, everybody, for joining, and I look forward to sharing my work. To get started, as I pull up my slides, I'm a professor at UCSF in the Department of Bioengineering, more importantly, in the program of bioengineering, which is joint between UC and SF and UC Berkeley. And I'm going to approach the work I present today, which is something that I've spent a good part of the last decade and a half focusing on, despite the fact that I don't have any formal kidney training. It's really driven by collaborators in the clinic who drew me in, collaborators on the engineering side who supported the work we do. And I think the message here is really try to bring together different groups of people together to advance this work. So let me go ahead and put my slides. Let me know if we're there.
Yes, we can see.
Okay. So terrific. So I actually, titled this after talking with Abe, Reimagining Kidney Care. I'll talk about my work, but I think what's really important is that there's a crisis that's growing, just not in this country, but worldwide. And that's the crisis of chronic kidney disease. And this is a disease that affects close to 1 billion people in the world. Think about that. 1 billion people in the world, over 39 million people in this country, costs us in healthcare costs of almost $150 billion annually. And if you look at this number of 843 that I have on the screen, it's actually more than diabetes and hypertension, and cancer combined. But we never pay sort of the attention to this disease that we might pay to diabetes or to cancer or to heart disease. And it's growing, driven by a number of factors, but driven by the fact, in addition to the conditions of diabetes and hypertension, which are the significant underlying causes, there are other genetic causes as well. So it's a large burden. The World Health Organization has started looking at this as one of the top 10 killers of people worldwide. So when you think of chronic kidney disease, it's 1 billion, and 40 million people in the US are affected. But the real impact, where it becomes fatal, is when you go to something called stage 5. And when you go to stage 5, you really don't have an option unless you get a transplant or you have to go on dialysis. So the chart on the right basically is how these different stages of CKD are defined. And the clinical determination is something called glomerular filtration rate, or GFR. So if you get to a GFR of less than 15 milliliters per minute, it basically means your body is not clearing toxins fast enough. Anywhere between 10 to 15, you can be listed for a transplant. It's rare that if you're below 10, you don't get on dialysis or a transplant. And these are the two solutions that have been around for the last 50 years. So, and what is interesting is this end stage kidney disease population has been growing. So what you're seeing is statistics for the last 25 years or so. And what you see is a steady increase. Despite all the improvements we as society in this country have brought to bear on chronic disease. I think it's interesting to see how it grows up, and then you have a knee right around this part. And people are like, wow, gosh, what did we do? We didn't do much. People died because of COVID. So one of the most vulnerable populations during COVID were kidney patients. In fact, I think one of the very first kidney patients that died in California was a dialysis patient. Even without a kidney transplant, you're immune compromised. So what happened is the prevalent population decreased because of COVID. So what are we talking in terms of numbers? These are big numbers, okay? We have 835,000 people with ESRD in the United States. But it turns out about 550,000 or so are being treated by dialysis. And there are two kinds of dialysis, hemodialysis and peritoneal dialysis. The vast majority are treated by hemodialysis. And for the people who are not familiar, if you go driving around, you might see centers for dialysis, DaVita, Satellite, U.S. Renal Association. Those are centers where patients go and get treated. Peritoneal dialysis is actually something patients do at home, but as you can see, the numbers are very low. And there's some interesting reasons for that. And the numbers of transplant have leveled up around 25,000 or so. Despite all the social engineering we're doing, trying to encourage people to donate via DMV, we have social media. In fact, if you actually mention on your social media that you might be interested in doing a transplant, they actually capture it somewhere and it's sent out to the patient advocacy organizations, the transplant advocacy organizations, so they can then reach out to you. So these numbers are going up, but you can see that the numbers of transplant are still significantly less. And the costs to Medicare are $50 billion. Now, that doesn't necessarily mean much. The NIH budget is smaller than $50 billion, and this $50 billion is just for treatment. It's not about the work, salaries lost because they couldn't work or productivity loss. This is actually what Medicare pays to treat these patients. And for people who are interested, most of the patients who are gone dialysis don't work. Most of them on Social Security. So I think another interesting thing is the mortality of patients on dialysis, especially hemodialysis. Here are two graphs, and I compare patients on dialysis with respect to cancers. And except for lung cancer and pancreatic cancer, most other cancers allow you to survive more. Dialysis has a mortality of north of 50% after 5 years. Think about that. You could get breast cancer, prostate cancer, and have a higher chance of survival. And that's because of all the investments we've made in cancer diagnostics and therapies. And immunotherapy, as many of you know, is changing the world. But unfortunately, dialysis has had this high mortality for many, many years. Now, I'm going to summarize the population of people on ESRD. So when I talk of ESRD, I think of both, we should talk about both the patients with kidney transplant and the ones on dialysis. So one, if you get a kidney transplant, which is like a lottery because only about 25,000 kidney transplants are done every year, and we have about 100,000 people on the wait list. Now remember, 100,000 people are not all the people doing dialysis. There's about 500,000. So you have to be sickest of the sick, but not too sick, and you have to be in the right place to get on the transplant list and get a transplant. So it's winning a lottery. But if you do, your survival rates are pretty good. 5-year survival, north of 80%. 1-year survival, close to 100%. Dialysis, look at the 5-year survival, very low. The quality of life for dialysis, most patients just don't work. Most of them come back from the clinic and go to sleep. We as engineers and scientists sometimes do not appreciate that, but over the years I've gotten a chance to talk to the patients and patient groups, and that's one of the biggest things that they point out to me is that the fact that it's not just about going to the clinic. It's the time it takes to go there. You get the treatment 3 to 4 hours. You come back, you feel wiped out. Now remember, I said many of these patients have hypertension as one of the leading causes of the kidney failure. Well, they have hypertension, but when they go into dialysis, they come back suffering from hypotension because the process of dialysis, where they take blood out of your body through a filter in 3 to 4 hours, trying to provide the amount of clearance that you need for a whole day in that 3 to 4 hours, is actually relatively physiologically challenging. So many of the patients experience low blood pressures. So they come home, sleep. The next day, they're miserable, and the following day they go back. So only about 10% of the patients work. And where do the costs come in? On the average, every dialysis patient is admitted to the hospital 2 times a year. So these are all statistics, but these are things that affect life, generate costs. So fundamentally, we've got two challenges: transplant and dialysis. Transplant is scarce. And if you're lucky to have it, you have to get an immunosuppression drugs, which have their own side effects. So on the average, transplants are rated for about 10 years. It's some people go longer, some people go shorter, but it's an average lifetime of about 10 years. And the drugs that you give for the transplant so that the patient doesn't reject it, not only do they have the side effects of making you less susceptible or more susceptible to disease, but they're actually nephrotoxic, meaning they're actually affecting the kidney that you got. So I think over the last 10 years, there's been a growing realization that something must be done differently. And a number of groups, both within the country and outside, have started thinking about what I call next generation renal replacement technology. So the idea is you're going to provide replacement of renal function. So the standard hemodialysis is here, shown on the top left, and I call that the standard treatment because that's the vast majority of patients treated. So blood is taken out from your vein through a semi-permeable filter, and you remove uremic toxins, and you do this for 3 to 4 hours. Then you've got the peritoneal dialysis on E, which is another standard option. So people are talking about how can we do better? So people are trying to say, can we actually make dialysis more continuous? Can we provide it such that patients can do it more frequently? And there is some evidence to say if you can make it more frequent, there are benefits. And if you can do it every day, and there have been clinical trials that have shown that, if you can do it every day for 8 hours a day, which is actually practically hard to implement, but if you're able to do it, you actually can have significant health benefits. So there are groups that are working on portable or wearable hemodialysis. And there's a groups that are working on implanted dialysis. Then others are working on wearable peritoneal dialysis. And then a couple of groups, including us, are working on what I have here, D, an implantable artificial kidney with the idea that we want to leapfrog the challenges associated with dialysis and provide something that's more akin to a kidney transplant. So that's the landscape. And in California, I would say there are companies in SoCal that are working on wearable and portable dialysis. There are companies that are working on next generation filters. And then of course there's work, I'll touch about this later, people who are working on using regenerative medicine. So people are trying to grow a kidney. People are trying to regenerate kidneys. But those are not really focused efforts. And I think, as I'll allude to later, maybe those are opportunities because they're underserved areas of research. Maybe this is an opportunity for us as UC scientists and engineers. So maybe at this point, let's just talk a little bit how do kidneys work and how do dialyzers work. So going back to our fundamental physics and principles of transport, we've got two ways you can remove solutes from blood. You've got a convective principle where you basically use pressure to drive out small solutes, whether driving out the blood molecules or components of blood. And it's a pressure-driven filtration. And the pressure-driven filtration, by first order, all the solutes are removed at about the same rate. I contrast this with diffusive transport, which I show on the second part here, where we're really relying on osmotic gradient to drive the solutes out. So the larger molecules come out slowly. The smaller molecules come out faster because of diffusivity, but also it's driven by the oncotic gradient. And the reason I show this is in our native kidneys, we operate on the basis of a convection. It's a pressure-driven filtration. Most dialysis that we do in the clinic is based on diffusion. So one of the reasons dialysis has the complications it does is because it doesn't really remove toxins in a physiological way. In fact, some of the larger molecules which are toxic don't get removed very well, and they continue accumulating in the body, and that leads to complications. And the reason we don't do convective filtration in dialysis clinics is because of the amount of water that would be needed, because you have to replace that water. I'll tell you what I mean by that in a second. So the vast majority of clinics in California, in the country, rely on diffusion. How much water? Each dialysis session, guys, requires about 120 liters of dialysate solution. We're in California, we care about water, okay? That solution is dumped down the drain. So 120 liters, say 40 gallons per sessions, okay, is dumped down the drain. And to generate that dialysate, which from RO water, you need about 3 or 4x. So just look at the back of the sustainability equation. And that's probably another opportunity for us as Californians to work on this. So this illustrates how our kidneys work. The kidney nephron is the functional unit of how the uremic toxins are cleared. So I've simplified this from an engineering perspective. You've got the filtration unit, the glomerulus, which I call the hemofilter. In a healthy kidney, we filter and generate about 150 liters of filtrate. So our blood is getting processed. We generate 150 liters. But when we go to the bathroom, we only pass out 1 to 2 liters of urine. Why is that? Because the rest of the kidney's nephron selectively reabsorbs much of that water and some of the valuable small solutes like glucose and small proteins, and concentrates the uremic toxins into 1 to 2 liters of fluid, and that's urine. So that's what I mean down here. I'm calling it the bioreactor. So about 99% of the filtered water is reabsorbed. If we did not do that, we'd dehydrate. So that's why we do not dehydrate despite the fact that we're removing 150 liters of, generating 150 liters of filtrate. Now, in dialysis, we do not actually do this. We do not remove we just use a solution and use an oncotic gradient to draw the toxins out. If we use convective principles, we'd have to replace most of this 150 liters, which becomes impractical. Now, there are ways to think about, how to address that, and that's an interesting area of research. But by and far, my point here is that one, the kidneys operate by convection. Second, the tubule does a number of different functions. And I'm just down selecting to this one very key function of volume reabsorption. Some of you who are familiar with kidney physiology know that kidneys are responsible for making sure that our vitamin D gets activated. We help with blood pressure control. There is the production of EPO, red blood, the hormone that stimulates red blood cells. There's actually a list I have in some other presentations of 7 to 8 different functions the kidney does. And when you go on dialysis, you lose obviously all that, and maybe you just do the filtration function. And dialysis does a filtration function just above the 10%, so that you're tethering between not enough function and just sufficient function. So with that stage, our goal when I took on this work was, let's see if we can build something that brings the principles of engineering to create something that's similar to transplant that mimics the nephron. And that's the vision that led to this cartoon. So what you're seeing here is a completely implantable device. And conceptually, it's got the filter, the hemofilter, which mimics the glomerulus, and another compartment that I call this bioreactor that mimics the tubule of the kidney. And it will be implanted into the abdominal cavity, similar to how a transplant is done. And again, if we start from what the end goal is, we want continuous treatment. Remember, I said there is some evidence that if you do more dialysis is better. If you do transplant, it's the best. So continuous treatment is a desirable goal. The second, from a quality of life perspective, is freedom of mobility, which basically means that patients don't have to go to the center to be tethered to a machine, but they can actually travel out of town. Third, by putting it all inside, and as we've learned from other disciplines where they have catheters coming out of the body, you minimize the infection risk. When you have catheters coming through the skin, that's a failure point. For people who have done ventricular assist devices, aka artificial hearts, they have drive lines, electrical power. That's again another infection risk. By having a combination of the tubular function in the bioreactor with the filtration function of the glomerulus, we provide more physiological therapy. So this should lead to presumably better function than dialysis. And based on a lot of the patient feedback and clinician feedback, if we can design our system such that we can minimize, ideally eliminate immunosuppression, that's a big step, because it not only avoids the side effects, there's a cost benefit too. And going aside to a little bit of public policy about, this has just recently changed, but for the longest time, if you're in dialysis and you're a Medicare patient, that you're covered. And interestingly, all dialysis patients in the US, no matter what age you are covered by Medicare. So, okay, you're getting dialysis. If you're lucky enough to get a transplant, it used to be just until last year, you only get immune suppression coverage by Medicare for 3 years. So after 3 years, if Medicare will not pay for your immune suppression medications. And so what that basically means is if you didn't have private insurance, and you couldn't afford immune suppression, you basically lost your kidney. And this is actually a real issue, as I found out from talking to patients who basically say, I got my transplant kidney, I was lucky, but after 3 years, I couldn't maintain maintain it because I couldn't afford the medication. What are we talking? It's not $100. We're talking about almost few $100, a month. Sometimes $1,000 or more a month. So it's significant cost. This has recently been changed by a lot of advocacy from the patient group. So that's a good thing. So anyway, so this is our goal. So the question for me and for you is, well, it's a lofty goal, but is there any basis for this? And it turns out there is. So almost 25 years ago, there was an effort at the University of Michigan, and we got inspired by it. They created something called the renal assist device. It made it to clinical trials. It was just an investigational device. And what this group did was to take standard off-the-shelf dialyzer technology, and that's what you see on the center, but connected in the architecture of the nephron, which I show on the left. The first stage is again a standard dialyzer cartridge, and dialyzer cartridges are just filters. And the second cartridge, they're lined with kidney cells. So what they did, the first one, again, operated in this convective filtration mode, where they have generate filtrate through the dialysis filter. And then they passed that filtrate through a second cartridge into which they had put cadaver kidney cells. And this was a research project. It made it to a clinical study, and you're seeing one of the hospital rooms where this was being tested on a patient when this picture was taken. And I label the filter here for blood filtration, the bioreactor, and this little beaker on top of the scale is where the urine is. And all the rest of this is the hardware to drive blood, okay, and replacement fluid, what I'm called, because they're generating a lot of ultrafiltrate. But it was exciting because they're able to show in their clinical trials on a cohort of patients who were suffering from something called acute kidney failure. So this is different. This is basically people who have been in trauma, their kidneys have shut down, and the standard therapy is the intensive dialysis in the hospital. Often that did not work. So this group was looking at ways to address that, but they found that by combining cell therapy, which is the bioreactor, with the filter, they're able to get to 50% mortality reduction because of the cells. So that inspired us to think we can actually bring our engineering toolkit and maybe build on the work that was done there. As you can see, this whole system is very large and unwieldy, which basically means it's not going to be suitable for chronic kidney failure, the patients that have to do this 3 or 4 times a week. So at this point, we put on our engineering hats and said, let's see how we can think about miniaturizing this. And it's instructive to look at what a hollow fiber dialyzer cartridge contains. It contains these tubes that are called the hollow fiber filters. So these are cylindrical tubes. The blood goes in the center, and the walls are porous. The walls are the filter. And what I've been my tortuous polydispersed pores, basically they are filter holes. They're not that different sizes. They have tortuous paths. And as an engineer, you can sort of analyze this and think about different pros and cons. But the point is, this is the hollow fiber, and this is what is the workhorse of filtration in a dialyzer. And if you contrast this to what we have in our kidneys, it's quite different. So in our kidneys, and I'm showing a histological image, a scanning electron micrograph of the glomerulus, and a transmission electron micrograph. What you have is basically in the kidneys, you've got in the glomerulus uniform slit pores. They're basically also highly uniform in size, but they're not like round circular pores. They're long, narrow pores, and they're about 5 to 10 nanometers, depending which TEM you look at. But the point is this here on the picture at the bottom, this is the endothelium, and this is an endothelial cell. This is the opening, and this is the basement membrane. And what you're seeing here are the foot processes of some cells called the podocytes. In the center, you're seeing the scanning electron micrograph of the podocytes. So it's the space between the podocytes that is the fine filtration unit. And if you look into this, like other people have done, there's a lot of chemical moieties that also provide not just a size selectivity, but also a charge selectivity. So you've got heparin sulfate that provides a little bit of charge selectivity, and most proteins have a negative charge. Heparin sulfate has a negative charge, so there's actually some repulsion as well. It's a very elegant filter. It's only the basement membrane part is just on the order of a fraction of a micron. So it's nothing like what this membrane looks like. So if we're going to build this device into something that's wearable and implantable, I thought as well, maybe we should start by trying to make something that mimics the kidneys filter. And the kidneys filter just operate on convection based on blood pressure. So it is at this point that, I went back to my engineering training and said, well, there's actually a technology that can help us get to feature sizes down to the order of the basement membrane. And that's the work in semiconductor microfabrication and nanotechnology. And this is the same technology that's been established. Today, we're able to make features down to 1 to 2 nanometers with precision. So we adapted that technology and we've been able to create a new membrane technology platform that we call the silicon nanopore membrane. What this is basically using silicon wafers and doing techniques of lithography, etching, depositions. And here you're seeing a silicon wafer on the left, scanning electron micrograph from the top, and on the right is a cross section. But you can engineer them so that you can control the size, the thinness, and make this in bulk. So you can get high selectivity, and if you make it thin enough, lower fluidic resistance, and you can make them in batch, which basically means you can make them reproducibly, but also potentially in cost-effective manner if this went ever to production. Here are some examples of scanning electron micrographs from some of our different runs. You're seeing, over the years, we've built different generations, different designs, and you're seeing some of the burst pressures are very high, more than what your car tire pressure is. Just to give you a sense, in our body, our pressures are probably on the order of a few PSI. Okay. If you're thinking about 200 millimeter bar blood pressure, that's very high. That's like 4 psi. So anyway, we've been working on the membrane, silicon membrane. We've brought a lot of engineering principles. How do you make them robust? They have to be very thin. They have to, be able to provide the surface that will allow blood to flow. So you're seeing some of the different variations, and again, hierarchical design principles we can get to. And the pore sizes we can go down to, just a few nanometers. And how big is a few nanometers? Well, albumin is about 7 nanometers in diameter, okay, to give you a sense. And albumin has a molecular weight of about 66 kilodaltons. Okay, so then here what you see on the right-hand side is a cartoon showing, the pore size of ours versus others, and we have a sharp cutoff, and we can tune it so that we can pretty much remove all the small solutes like a glomerular basement membrane does, but still preserve albumin. And of course, don't let red blood cells through, and also prevent antibodies from going through it. So that is the cartoon. On the left-hand side, scanning electron micrographs of the filter on the very left, a polysulfone hollow fiber, that's the one I showed you before. It's 35 microns, and our membrane, which is half a micron. Just the back of the envelope tells you that the resistance is going to be, well, let's just say 70 times less because for everything given being equal for our membrane. And then by using lithography, we can space the membranes very closely together and get, in this case, 7 nanometer pore. The parameter we use to analyze the permeability is something called hydraulic permeability. And we have normalized it. Our membranes have hydraulic permeability an order of magnitude. What does that mean? You do not need a large pump. You could presumably, and we've shown this, just use cardiac pressure to drive the filtration. And because you have a sharp cutoff membrane, you can remove the small solutes as well as the large solutes with efficiency. And we're able to show that here and also show that we don't leak albumin and also allow small solutes and toxins to go through. Here you can see some of the cytokines, the glucose and insulin going through. And by tuning the pore, you can just control what you let through or not. Interestingly, we don't have any passage of albumin, but also we don't allow any antibodies to go through, which means this membrane could potentially act as a steric barrier for immunoprotection between the patient and any transplanted cells. So this was promising data. And then the question that usually comes up when you have a foreign material is, well, how do you deal with blood contact? Blood contact is a challenge. You're bringing a foreign material. There's a whole coagulation cascade we'll have to deal with. Absolutely true. So we started looking around and talking to collaborators. And again, as an engineer, we are collaborators. We look around. We don't have to invent everything. And there's been so much um, uh, research that's been done on biomaterials. So I actually had a lot of candidates materials to choose from. And then as I started looking at it, we found that while the materials were very blood compatible, as done by other researchers, they may not necessarily work for us in the way they're developed because many of the materials are designed to be used for, stents, heart valves, etc., where there's no need for filtration. In our case, we have 10 nanometers or so for our filters to prevent albumin leakage and allow ultrafiltration. So if we took many of these coatings and slapped them on, would block our filters. So that didn't work. Then, unlike heart valves or stents, we're in a different flow regime. We're in generally much lower flow regions of like low shear and low flow rate. So we had to come up with ways to think about how do we minimize the protein fouling that will occur in the filters, but also formation of thrombi. And the approach we took is techniques of self-assembly and layer by layer assembly techniques, such that you can coat our membranes with 1 to 2 molecules thick. So here's some early data from where we looked at different coatings. In this particular case, we looked at something called polysulfobetane methacrylate, a zwitterionic polymer, and showed that by coating these membranes, you are able to have just 1 to 2 molecules thick, and you can get very strong resistance to protein fouling and also prevention of any thrombi. And that's what the SCM on the right hand shows. So we took some unmodified silicon material and you can see cellular debris. On the bottom, this is the coated with a zwitterionic polymer, put it into a model, animal model of high thrombogenicity. It's a pig model, and left it in for a month. And we're getting this material out almost pristine. So now, with what was exciting for many of my colleagues in the clinical side were like, well, you actually, if this is feasible, not only do you prevent thrombosis on the filter and prevent its failure, but you are actually allowing us to think of avoiding the administration of blood thinners. Anytime you have a stent, anytime you have a heart valve, you have to have blood thinners, which have their own side effects. So by eliminating the need for blood thinners, we actually may be providing an additional benefit. So far I've talked about how do we improve the membrane, both in terms of selectivity, permeability, and now in terms of blood compatibility. Well, our device is not just the membrane, we also have to have cells. So there's a number of different ways to look at the cellular component and the sourcing of the cells. So here is where I had to lean on collaborators and also talk to the FDA. And the FDA said, obviously you can come to us with any cell that you want, but you have to do a number of tests. And the only experience the FDA had until the time we went there was the work that was done by that group I showed you from the University of Michigan. And then it used human cadaver cells. So we said, okay, we'll work with that. We're also working with pig cells. We're working with other animal cells. We're also working with human cell lines. But we said, okay, if that's what it is, let's focus on using the human renal tubule cell. So working with my colleagues, we're able to develop protocols, and these are different lines showing how we're able to expand the cells from culture. So we ended up, there's a sort of what I call the standard protocol here in black. And you can expand the cells, but they don't expand very much. But using a protocol called EP here, enhanced protocol, you can actually grow them many times more. And the variation here is actually different donors. So this donor in green, who's highly prolific, happened to be a very healthy donor, tragically young, accident victim, but the kidneys were very healthy. Whereas some of the others or older cancer patients, whatnot. So there's a variability, my point. But the other point I'm making also is that you can use human renal tubule cells, and we focused on that initially for FDA purposes. But that's not the only way going forward. So we took this and said, okay, do they actually behave as we should? So among other things, the human renal tubule cells have to be able to transport albumin. And we are able to show that, yeah, they do not only transport albumin, but they respond as a function of flow, as they should. We're able to show that they express the biomarkers that are typical of the proximal tubule, and they do respond again to shear stress. OSS stands for orbital shear stress. And if it's static, they behave differently. Now, my colleagues who do biology research sometimes get excited by this work because now we're actually mimicking what happens in the kidneys, and much of the kidney literature in the past has been based on static studies. So my point is, as bioengineers and biomedical engineers, we've now created a tool that not only is good for us, but we can also share with the biological community so they can do additional experiments. But these biomarkers are basically markers of health or markers of transport. NHE3 is a marker of sodium transport. That also mimics the amount of water transport that goes through. The 1A hydroxylase is a production of vitamin D, which is a precursor. And then therefore, you basically know that these cells are behaving in the proximal tubule. We've also shown that mechanobiology does affect it, and there's actually some unexpected effects. So when we grow cells on a tissue culture dish, it's usually shown in the bottom here, the 40 kilopascal, after a week or so, they get to confluence. If in a week, if you grow on a very soft substrate, it doesn't really see, nothing seems to happen. So most of us who do experiments in the lab, we'll usually do it in the tissue culture polystyrene and get, oh, here's a confluent cell line, and not really pay attention to the one at the top. But look at what happens after 4 weeks. The one at the top actually started expressing the NHE transporter, that vital biomarker of water transport that's unique to kidneys, almost 25-fold. So and this is an unexpected discovery that says, look, you can actually get to a biomimetic function, but you have to work with the conditions that the body has, which is soft tissues, but you have to culture it longer. We've taken this, and here you can see on the right-hand side, that they also have the biomarkers of function of mechanotransduction, which is the cilia that these cells have. So this was exciting, and with this protocol, we've been able to keep the cells in the incubator going for almost a year. So we know these cells can maintain phenotype if you feed them right. But as I said, one of the things we want to do is to actually transport water. Remember the filtrate? So here we actually looked at, can we modulate the water transport by using hormones? Can we actually disrupt it? So it's not people can say, well, you have a leaky barrier, so water is going to just leak through. So you can actually use something called obane that can inhibit active transport. It inhibits that active transport of water through the membrane. And when you put obane, you can see the amount of water transport decreases. If you put if you increase the oncotic gradient, which is also in our body, the blood has the oncotic gradient between the blood at capillary and the tubule, you actually increase. So, and you can see mannitol can also modify the gradient. And you can also put other hormones that increase it. So by doing this, we said, hey, look, it is possible, at least with human cells, to get up to 13 liters per day per square meter. But it's not only human cells. You can do this also with animal cells. So here we took this animal cell line, and we're able to show that, again, we can also increase or modulate the water transport as 4 times higher, 5 times higher. So my point is, there's actually a lot of research and optimization and ways to get to more tubular-like cells. And here on the right-hand side is showing that actually the flow rate is a function of shear. So we're getting to this regime of 70 liters per day per square meter, and you do the back of the envelope math, you're going to do something that eventually can be the size that we want to be able to put in. So, okay, so we have this proof of concept experiments. So now I'm going to go right through the engineering side. Okay, now we've got basic sort of like science. Let's see if we can actually make this into something that works. We made a prototype filter to say that, does this work? Can we actually make a device that we can implant and don't put any blood thinners and show that it doesn't clot? And here is one of our early prototypes, and we're able to show that just using aspirin, no heparin, no coumadin, no warfarin, we're able to get, you know, patency, meaning there's no need, the blood will flow through without clotting for about a month. A lot of this is again engineering. You have to move design for laminarity. You have to design for optimal shear stress. You have to take into account that you're moving from blood that's circular, coming from a circular vessel, going to a parallel plate system, and some engineering optimization that happens. Then we actually looked at clearance. We always have this assay in our lab where we test in vivo for 3 days, and we're able to show that in this animal patient here, just saying, again, no blood thinners. There's no loss of albumin. We're able to remove creatinine and phosphorus and other small solutes pretty normally. Yeah, initially there's a little bit of a bump and then it stabilized. So we felt like, okay, we've got a filter that seems to be visible. Then we said, okay, let's look at the cells. Again, use the same. Let's integrate kidney cells into this. And the challenge we said is let's put kidney cells, but from humans and test it in a pig. That way we can test if this immunoprotective concept seems promising. So what you're seeing here is the device, again, with each of the little squares are kidney cells, where the earth where kidney cells are. And you get connected to the vessels in the neck of the pig. And this we learned because we could monitor through ultrasound and left it again for 3 days, sometimes 7 days. And we were able to show that, again, without immune suppression, without blood thinners, we saw evidence of protection. Compared to the cells on the incubator, they're the same. So we had controls in the incubator that are not obviously implanted. They had the same level of viability. They expressed the biomarkers of function, the sodium hydroxyl, hydrogen transporter, the vitamin D, and aquaporin, which is a water transporter. On the right-hand side, these are biomarkers. One is of health, GGT activity. One is of injury. This NAG activity is a biomarker for kidney injury. Like if the cells are being challenged and not happy, we'll see a problem. In all cases, you can see relative to the positive control, the implant does pretty well. So for first order principle, we've got now a bioreactor that not only functions but also provides immunoprotection. So we took this and said, okay, what can we do to show feasibility? So we said, let's integrate the prototype filter with the prototype hemofilter in the architecture that those guys in Michigan and then the architecture of the nephron. So we connected it here together. It's about the size of 2 decks of cards. And then we brought out my surgery colleagues. And what you can see is they're putting this in. So this is the filter. Here's a sleeve that protects the connectors. And here are the ringed grafts that go to the iliac vessels. These are the same vessels that you do in a transplant. And the little purple tube is what goes to the bladder. So let me see my video play. Yeah. So what you're seeing is the video of the very first surgery of us putting it in. The clamps have been taken off, and through the device you can see it's red. Blood is getting perfused just based on cardiac pressure. And this purple is what's going to get connected to the bladder. It has not been connected yet. So they're just making sure it works, and they do. And here we go. See that we're coming back red again. No blood thinner, no immune suppression. And after this, it's put back in and it's closed. And we always say, if it worked for 3 days, we feel it's a good first step. So that's what we did. And then, oops, sorry, hold on, I'm going to go back. Oops, okay, here. Right before we explanted the whole device, we took the connection off the bladder. So this tube was going to the bladder, and what you're seeing is clear ultrafiltrate. This is urine. I'm going to call it urine, proto-urine, because it doesn't have all this, we don't have all the different types of cells. We only picked one type of cell to go in. But the point is, we are creating a clear filtrate that has no albumin, has the concentration of urine. And when we analyze this, we find that the cells were viable, despite we had, in the pig, despite no immunosuppression. And we had 95% reabsorption. Remember, I said the reabsorption rate is about 99%. So we're very much in that ballpark of what we think. Now, I'll just preface this as scientists. This is preliminary work. We have to do this again. We have to analyze it more, etc., etc. But this is the first evidence that a combining cell therapy with a biological filter, without immunosuppression, without blood thinners, we're able to create something that creates urine at a percentage that's similar to what we'd need. So I'll summarize by saying, what have we done? We've taken a hybrid approach. Okay. We've tried to incorporate biological metabolism into our system by having renal tubule cells integrated with the silicon membranes. And the functional proof is no electrical power, it is totally implanted, no systemic anticoagulation, no immune suppression. So this work is really sort of at an inflection point of looking. We need to now move to the next level, which is more evidence to establish before we can go to human study. So I'm going to wrap up by saying, this is not just me. There's a team. I have a clinical partner who helps me think through these things, lots of supporters. But I'd wrap up by saying a little bit about California. So here are the statistics of California. We have almost 6.7 million patients in California with kidney disease. Okay. Look at those numbers. We have 81,000 patients on dialysis. Okay. Now remember, each patient is about $100,000 to Medicare. So you can do the math. We have close to 28,000 people with a kidney transplant. Um, this is not California. I should say, this is in the country. Okay. In California, there's 18,000 people on the wait list. Okay. So, no, I'm sorry. This is the transplant number of patients in California. I'm confusing this. In the country, there's 250,000, so 10 times that amount. The wait list in California is 18,000. If you're in California in the wait list, you're on the wait list on the average of 10 years, which means this the game is against you because most people die within 5 years. And the highest burden, as you can see, are some of these inland underserved populations, and there's a big equity gap. The black population is 4 times as likely to suffer from kidney failure as whites. Hispanic populations about 3 times, and even Asians, 30% more. And access to dialysis, despite its life-saving and even the challenges, it's hard. If you go in you have to drive 30, 40, 50 miles to get a dialysis center. So I think despite the fact that we have these services available and for the last 50 years, we have a huge burden in California. And I'm thinking, I'm taking one stab at this, but there's a way for us to maybe all think together as UC, all of you, students, postdocs, faculty, clinicians, that maybe we should think about how can we address this across the system. And we should think about this together because we actually have a unique opportunity as a public land grant university to do something for the state of California as well as for the nation. And we have the talent. But what we haven't done is focused ourselves. So when Abe and I talked, I said, Abe, this is not just about talking about research. We actually, as the Bioengineering Institute of California, have an opportunity to take something that nobody else is addressing in a meaningful way and bring in other people. It's not just about building an artificial kidney, better diagnostics, better delivery of care. Use AI to identify patients that can be benefit from different therapies. Make the research in stem cells focus on kidney. Focus on xenotransplants. So there's a whole host of opportunities, and I am not saying, it's my way. My way is one way, but we can collectively bring all of us together and make a unique kidney initiative based at the University of California. So that's my spiel, and thank you, everybody, and thank you for uh, giving me attention.
Thank you. Thank you so much. That's such a comprehensive, fantastic coverage that I think everyone can relate to and understand. So I think before we get into Q&A, I just want to ask our web master guru, or on our Zoom guru, Gisela,
Yeah.
do we have over 3 minutes to have, because I have
Yeah,
so many
yeah.
questions. Can we
Yeah,
go
we have
over
time a little bit. Yeah.
for those that can?
Hmm.
Okay, sounds good. All right, so let's open up for discussion, questions, and anything on your mind that might be related to what we just heard. Any questions? You can, I know we have lost some participants. I think it's early in the day, probably in midday, a lot of meetings come up, but
Hmm.
um, you could just turn on your mic or put a question in the chat. If not, I'll go ahead. I have quite a few questions, but I'll start with a few. I guess first question, Shiva, just wondering, the dialysis centers, are there efforts to do dialysis at home or some kind of hybrid, meaning doing, every so often you go in and but you can do a lot of it at home. My friend, as I mentioned to you before, he was my elementary school friend, and he's doing 3 dialysis a week, and I can see many many hours that those 3 days, and then also the recovery, the fatigue, and all that.
Absolutely right. So if you see here, bottom here, there's something called home hemodialysis or peritoneal dialysis. Those are both done at home.
Oh,
And the numbers are very low. I mean, look down here, okay, at the very bottom. So we have less than 50,000, we have 500,000 patients on dialysis, less than 10% do it at home. Now, what is interesting is there's actually a lot of evidence that says if you do dialysis at home, you feel better and you actually do better. Not only feel better, but your actually health is better. But for a long time, it was not done. And it was not done because of economic you know, incentives or lack thereof. So at one point, they used to pay you more if you did dialysis in the center than if you did at home. That
hmm.
has changed, and they're trying to encourage now more people to start at home. So there's two types of home dialysis. There's peritoneal dialysis, which is where you use your stomach, and the peritoneal membrane is the filter. And then there is hemodialysis, which is basically what you do in the clinic, but do it at home. Peritoneal dialysis, it's doable. The challenges are you have a higher risk of infection because the peritoneum gets infected, and then you have peritonitis, and that's all has all bad effects. So
Hmm.
there's some patients that can't do peritoneal dialysis. And then home hemodialysis is just complicated. You have to be able to put the needle into your arm. You have to maintain the machine. So there's less than 10,000 people doing home hemodialysis. But if you could do that, absolutely. So there's at least a couple of companies uh, based both in California that are trying to make better machines for home. So we'll see how that goes.
Hmm. Thank you. That's great. Yeah. And if no other I'll quickly just ask a few. Um, I was just trying to think comprehensively. You mentioned there's different stages in the chronic kidney disease. People also have comorbidities, and then you mentioned COVID being one where, immunocompromised um, kidney patients succumb more at higher rate. So I guess what I'm thinking is there are technologies for different stages. For example, how do we slow the progress? How do we maybe so at the slow the progress of the disease, so it doesn't get to the point where you need to do dialysis or have a artificial kidney, and some of those, just the whole linear, I mean, the aspect of going from a healthy person to slightly kidney failure, slightly kidney affected, and then eventually to kidney failure. That process, I wonder if you can comment on how, if that were to happen, would it be just isolated efforts to do different parts, or there could be some kind of a more of a, like we were trying to talk about here, a collective effort that that can address different parts, but also complement each other. I
Yeah,
don't.
I mean, you're right on. So one of the challenges why we don't have uh, maybe earlier detection or earlier signs for kidney disease because it's silent.
Hmm.
So most patients don't even know they have kidney disease and they don't screen for kidney disease. So you don't know it until you either go into the hospital and they do it as this sort of like your nephrologist suspects something is up and then will do it,
Hmm.
or you basically crash into the emergency room. So one of the things that I think that has been talked about at the national level, and I think by the physician, can we do some sort of earlier detection and non-invasive detection? So is it
Hmm.
possible to develop wearables that measure the biomarkers that can be easily done at home? So I think there's an opportunity to do that and make that part of the routine testing. The other one is, let's say you've detected you have kidney disease. You can absolutely slow it down. That one, you cannot usually reverse it, but you can slow it down and you can arrest it. And what you have to do is to hit the underlying cause. So if it's high blood pressure, you go on blood pressure reducing medications. If it's diabetes, you got to control your diabetes. Um, you have to also, if you've got a point where you're taking you have heart disease and your heart is not pumping well, so now you've got fluid buildup in your body, now your kidneys are under stress.
Hmm.
Well, then you have to control the amount of water you drink. So each of these have known interventions, but how you detect when you need those interventions and make the interventions easier on the patients, or what I'm going to call patient-centric, have yet to be sort of implemented. And I think that's the opportunity for bioengineering as a discipline, is we can make patient-centric solutions by building on the known science.
Hmm. Yeah.
So another one is compliance. If you're a transplant patient, you're taking sometimes 30, 40 drugs
Hmm.
a week, different drugs. And one of the things that I hear from the patients is, just keeping track is hard. And then I forget if I took it or not. So I'm thinking like with all our groups that think in terms of compliance, AI, think in terms of apps, we should be able to make something that's scientifically valid.
Yeah, that makes sense. Let's
Yeah.
think about that. There is a question here from
In the
Randolph.
chat. Hmm.
Yeah.
How close are
Um,
we to having a device that can realistically be used in patients in renal failure? Years, decades?
So let me answer the question in a couple. So I think the most promising thing that has happened in the last 2, 3 years is xenotransplant. And I said promising in the fact that we actually have a new intervention in that's moved to clinical trials. So it's not dialysis, it's not what I'm doing, but it's xenotransplant. So this is a pig kidney that actually is now going into clinical trials. So they've actually treated uh, I want to say 5 or maybe 6 patients so far with pig kidneys. Now, either patients have died or they've had to take it out for rejection, but there's one patient that still has it in. And I think through that, we're learning that it is possible for people to take pig kidneys and keep them, but they need very high levels of immune suppression. Now,
Mm.
how long has that taken to get to this place? It's taken about 3 decades because this was started in the early 90s.
mm,
Okay, so I just want to make sure that people understand this work going on. And I think we also have an opportunity as you see to think about how
Mhm.
can we make that field better. But if you're talking about our device, I think if Randall's asking, I think the way I answer that question is what is the roadmap to getting the work done so that the FDA will give us permission to test in a human? That's like a 3 to 4 year time frame. We have to scale up. We have to do all the tests you need for an investigational device or investigational therapy, go with that data. And then after that, it's a question of then, how do you go through all the scale up and manufacturing distribution? So when we have talked to different stakeholders in the government, in the industry, it's possible to take what we are doing to patients within the course of 5 to 6 years if there's sufficient resource around it. And that's always the big question. Do we have sufficient resource? It's not decades. It's not going to take decades because of the technology or the
Mhm.
science. It may take a decade because of organizing the funding around it.
Mm. Yeah, it's
Mhm.
always a tricky one.
Mhm. Cool. Hey,
Ian,
Shiva, really quick. What is the housing material for your device? That plastic.
Yeah, so here in the prototype, it's a
Mhm.
medical grade polycarbonate.
Okay.
And we got that inspiration from people who are making something called the Berlin heart for babies. So they use an artificial heart pump and they use
Mhm.
the medical grade plastic. And we did it because one, it was medical grade, and secondly, it's transparent.
Mhm.
So putting it together makes it a lot easier. But
Mhm.
one might envision that for long term would move to something like titanium housing for a
Yeah.
pacemaker.
Yeah.
mhm.
Okay. Great. Thanks.
Yeah.
Mhm.
Right? Are there any
Okay.
other
Any other
questions?
questions?
Hmm.
No, I don't think so.
Yeah. So,
Okay. I think we can wrap things up. Thanks, everyone. Thank you, Shiva. It was an excellent presentation. It's like,
how
really
can people,
loved it.
if they have, questions or they want to discuss about how to work with you or anything like that, how can they just email you?
Yeah, so yeah, my email is in the chat and it's also at the end of the slide presentation.
Yeah. It's
Yeah,
in the,
people can
Mhm.
reach out. Happy to talk. People can talk about whether it's my work or their work or how we can all collaborate. I think the
Hmm.
other thing I would encourage all of you guys, and especially if you're trainees, students, reach out also and go attend every city has some patient support group. Go sit in one of those. You'll hear stories, and then you as a student in bioengineering can think about how you might contribute. For me, I know personally that was actually very uh,
Mm.
inspirational because, and I'll share this. One of the patients came up to me and said, Dr. Roy, what we need is not a perfect kidney. We just need something to get us off dialysis because that is where we are really limited once we get on dialysis. So it actually reframed how I think about it. Is like if we had to think in terms of a minimum viable product,
Hmm.
we should think about what is it that gets people off dialysis as opposed to just going for the moonshot the very first time. So it's like, let's go into space, low Earth orbit, then go to the moon and then to Mars, right?
Mhm.
And so I think that's one of the things that I think the patient-centric uh, uh, focus can help us because it will ground us as to what we need to do. And some of the other patients have come and said, look, what we want, again, I think somebody has come with, it's just a medication management system for us. And yeah, I think now we have a number of when I say families, family members of these patients that have made apps to help them manage their medication. Okay. The other thing for people who are on dialysis is drinking water, and maybe most of us don't know. Drinking water is a challenge because if you drink water, you have to wait until you get to the dialysis gland to take out that water. You don't pee.
Mhm.
Mm.
But if you have water in your body, your heart has to work harder. So you actually are limited to how much you should be drinking water. So think about that. Thirst is a reflex. We feel thirsty. The only way to address that is drinking. And if just imagine you can drink because, hey, I've met my quota.
Mhm.
Mm.
And so that's another thing that we think about. What are some of the other side effects of transplant? Itching. Yeah, I mean, itching.
Mhm.
That's itching. These are like, people are like, these are things that bother us, but nobody even thinks of it as like, it's not a life or death situation.
Mm.
But these are things we should be thinking about. Like, how can we help them think that? I can think of not only, ointments, think of, are there electrical stimulation strategies to minimize that?
Mhm.
It's an open field because for a long time, engineers and nephrologists have not crossed paths. And for those of you who are still online, the American Society of Nephrology wants to encourage more engineers. In fact, if you're a PhD student, you can join the society for free. It's a clinical society. They have an annual meeting. You can join the society for free. You get access to all their resources, and you can also apply for fellowships for
Mhm.
Wow.
PhD students.
those and the students, yeah, great
And
opportunity.
postdocs too. Yeah.
Mhm.
And they're encouraging engineering. And I'm saying when I say engineering, they're really encouraging people from bioengineering to engage because they realize that there's been a gap in the discoveries and what they're getting to patients.
Mhm. Well, on that note, I think we have our homework set for us. But
Okay.
Shiva, thank you so much. So, Abe,
Yes.
any last announcements about the
No, it's
community?
excellent. I just want to, hopefully this is just the start of, I mean, hopefully many more discussions and ideas that will bounce around and hopefully realize, much more improvement for the patients.
That's right. Thank you,
Thank you,
Shiva.
everybody.
This was excellent.
Thank you,
All right.
Shubham. Thank you,
Thanks for everyone
everyone. So
attending.
we'll be
Bye.
Yep.
Bye.